graphic.
Let's move to the next speaker. And I'm talking about Hania Koury from Agilent, where she works as a high-resolution application specialist. We got in touch with Hania last year because she ran a demo on a set of samples prepared by my lab. And actually, really impressed by the work that she did on our samples, and also on the possibility that ion mobility is enabling on this time-of-flight platform. So I would like to hear more about this. So thank you, Hania, and please go ahead with your presentation.
Excellent. Thank you very much for the nice introduction. I hope you can hear me and see my my screen
yes,
well.
You can.
And thank you for the invitation to speak in this meeting for today. I will be presenting the different solutions that Agilent will show you and help you to get beautiful data like the previous results, presentation. So the workflows that I will show you would start even beyond the LCMS, even in the sample preparation with the assay map Bravo, where that could help you to have very good reproducibility with your sample preparation. I will focus on the lipidomic, but a lot of things are in common with other omic workflows, in this case, like the statistical analysis or even the identification. So the journey, as I said, it starts by from, in this case, post-sample preparation acquisition, and for the lipids, for the lipid annotation and the analysis after obtaining these results. Just to make sure that we are both on the same page, lipidomic workflows, if I may split them in a simplistic way, they could be into two divided workflows. The first one would be like shotgun lipidomics, which is most of the time infusion based and done on a triple quad. In general, it is for quantitative approach where you're based on the quantitation of a class of lipids. So it is interesting, it is fast, it is semi-quantitative in this case, and easy. Unfortunately, in this case, you cannot distinguish isomers with it. The other lipidomic workflow that I will focus on today would be the profiling lipidomics. So in this case, it would be you have very complex samples, tissues, or from plasma, and you would like to run them and simplify, if I may say, the samples a little bit more in order to be able to detect and quantify them. So there is a chromatographic method before based, and of course you would need high resolution mass spec in order to do that. So it can be lengthy, but it's worth the trouble, I would say, because you can get a little bit more information about the classes and annotation of the lipids in this case. So yeah, the workflow is, as I've said, it is in general, it is as depicted in this scheme over here. So the idea when you have a complex sample is to simplify it and then run the mass spec analysis on them with auto MSMS, data dependent or data independent. And if you have the ion mobility, you could run with ion mobility data on them. The bottleneck in any analysis with the omics is the identification. So I will show you Agilent tools and how we could help you with the lipid annotation, and you could work through that and run, create your own database and do the statistical analysis. So step one would be the separation, and as I said, the chromatography. There are different types of chromatography. You can have normal phase, reverse phase. So like in the previous presentation, there was a question about ceramides, hexaceramides, and how to be able to separate them. There is another class of separation using supercritical fluid chromatography, where you're using carbon dioxide in its liquid state as a solvent, as a mobile phase in this case, can be used as well. In a way, I would compare it to a normal phase chromatography in terms of the separation. It can cover up a good class of lipids in this case as the LCMS as well. So it is something to think about sometimes when you have a question and you have a certain a project that you'd like to run, how to use as a separation. SFC can be used as well for lipid, and it works very nicely. Bearing in mind that normal phase separation, when used on complex samples, you're separating the lipids by classes. So you've got clusters of classes that elute in your sample. In a similar way, the SFC, since CO2 has same polar as hexane, so it works as a normal phase. So you are separating as well lipids by classes using SFC. A good thing with the SFC, a trick that we have used, and you can see this in a poster and different application notes in this case, is since you're using CO2 as in the mobile phase, so the pressure is very low, so you could couple two columns one after the other sequentially. So these results that I'm showing you here were done by one of my colleagues, and she has used a helic followed by a C18 column for this type of separation, and you get more insight, more in depth about what's in your sample. And of course, good separation in this short time would mean you've got better peak capacity and less in source, space or competition in source and ion suppression. This is what we're used to often seeing would be a normal phase, reverse phase separation with lipids, where you are separating over here by hydrophobicity. So depending on the questions that we're asking, different groups have their own different methods that answer their questions. The message over here is a good separation method is important to be able to analyze the samples. And a good method, for example, over here, you could even get separation of isomers, baseline separation of certain isomers. These are, this example over here that I'm presenting, these are two standards that were purchased separately. And as you could see, we could be able to distinguish them easily by a good method of separation. So I've mentioned the SFC earlier, and I've said that within the beauty of the SFC that you don't get a lot of pressure in the system, back pressure. So you could put two columns, one after the other. But we could also use 2D-LC in this case in order to get a better in-depth identification. This is in our system. So with Agilent 2D system, you could get, we have different modes of using it, multiple hard cutting, or even a comprehensive 2D method. That works as well very well for complex samples and as a solution. This part is chromatography, but with ion mobility, when we're talking ion mobility, you could use a system of separation, LC or SFC, and the separation in this case takes place in the second order of magnitude. By putting an ion mobility in between the chromatography and mass spectrometry in this case, where the separation takes place in the order of 60 milliseconds, you are as well getting an example or something that looks like a 2D LC actually, but in this case, it's quicker, shorter, you're increasing the peak capacity and getting most of your data. Plus it adds the dimension that you can do, you could use this mobility data or the collisional cross-section data produced as another variant for the identification. So we talked about the separation, but the acquisition as well is as important. So regular lipid annotation, and this is where I would distinguish between identification and annotation. In order to have a complete identification for any compound, you would need the exact structure of it, and if possible, to compare it with a standard. Annotation is practically we have an incomplete identification based on what we have from information. With lipids, it's a little bit sophisticated because we've got more than 43,000 compounds in lipid blast, and commercially we have around 1,000 standards that are present. So this does not cover all the theoretical lipids present. So it's not easy, or it's not possible to do an identification by comparing to a standard. This is why we are happy to do that using an annotation with comparison with in silico. In order to do a complete annotation, or and in this case, we would like to collect information using MSMS or other technologies in order to be able to tell the class, the elemental composition, the position of double bonds, where they are, the fatty acids, etc. Most of this information can be acquired actually and obtained using MSMS spectra. This is another depiction of what I've just mentioned in the previous slide, showing the importance of checking what's the class of the lipid, would be from the polar head group or the head group. The position of the fatty acid groups helps, would be an information that it's necessary for the annotation. The position, which one is it, SN1, SN2, which fatty acid, is it saturated, unsaturated, the cis transposition, etc. The orientation of these lipids. So in order to get the maximum amount of information in a particular sample, when we are trying to annotate and perform mass MSMS or data dependent analysis, usually these software tools, a comprehensive data dependent LCMS analysis, gives you high quality MSMS data, but it's linked to how many you in an injection. Usually we program it to do the n number of compounds. So this would be, in order to get more information, this is where in the acquisition we've got the solution with iterative MSMS, where you are looking and creating automatically an exclusion list and injecting the sample more than once and looking at more in depth the data. So in the example over here, I'm showing you an iterative MSMS with the top 3 masses to fragment. And when you're doing 3 iterative MSMS on this sample, once the run has finished with the first 3, the software puts them in an exclusion list and starts with analyzing or performing MSMS on the next 3 high in abundance and so on. So the more you do, the more you dig deeper into the low abundance. As you can see on the left side, on the blue lines, they show you the number of annotated lipids that increase with the number of iterative MSMS. And this is compared to a conventional auto MSMS that is used. Of course, you reach a point where you start plateauing because you cannot get more information than that, or the sample or the data or the samples are very low in abundance. You cannot get good fragmentation out of them. In the middle, this is showing you the abundance, the log of the abundance after 3 iterative injection. As you see, we're going into the lower abundance in this case. The bottleneck is the annotation, or the, in this case, with Agilent's new tool for lipidomic analysis, Lipid Annotator, you can get more information about your sample. It works using a multiple scoring algorithm based on the lipid blast with modified, I would say, lipid blast. We've added the Avanti polar standards and other standards inside this database. It works with data dependent, data independent, and all ion modes of ion mobility data. So it works with all the different instruments Agilent has. It has different export formats, the regular CSV and the PCDL for your mass hunter databases. It is easy to use, and as you see, it is not very complicated in terms of format. Quite nicer view where you could see these are the annotated lipids in an example of a sample. You've got the M/Z, the y-axis, and the retention time. So it is nice to see how they are distributed in a chromatography. This can be a nice, quick, at a glance look of how did it go and where are certain lipids. You've got a pie chart with a percentage of how many of each of the classes were identified in your samples and the feature list. So that's the feature view tab, and the more important, I would say, is the nitty-gritty details of each of the identified lipids in a sample. So each line would correspond, of course, to one of the lipids. You've got when you have enough information with MS spectra, you can get the annotation, complete annotation up to the dominant constituent, and if it's not sufficient, the software is, I would say, cautious and gives you doesn't make it the data speak more than it is, so it gives you the summed composition. On the lower part of the software, you could see the comparison between the observed or experimental spectra with the in silico spectrum. So each of these peaks were found. These count in this case as 4 out of the, for example, 6 fragments in this case of the PC phosphatidylcholine that was identified. You've got the different candidates on this table. Of course, it gives you why that first one was the best candidate. In this case, as I said, the PC, we had 4 out of 6 of the fragments that correspond. The next ones were 1 out of 6, so that's why it was the highest hit for that sample. So as I said, this works with data independent, and ion mobility, all ions data. The whole point of this is to create your own database library from your real samples, real biological samples, and you could use it for targeted and untargeted approaches or workflows as you wish. Another important part besides the annotation of lipids is the statistical analysis. What does the data mean in this case? And as we know, different lipid classes, we cannot compare apples and oranges in this case. So with the Mass Profiler Professional tool, there is a specific workflow for Lipidomic where you could use it for normalization of the data, and the normalization would be using a class-dependent lipid normalization. So you would be using the Avanti Splash, for example, Lipidomics, and normalizing the PCs with the PCs, the PEs with the PEs, etc. The example I'm going to show you in the next slides is based, it's an application note. It is based on a previous publication where they have used acute myeloid leukemia cells and did a lipidomic studies on them. So in this study, these cells were treated with either MPA, which is medroxyprogesterone, or with bezafibrate, or even a combination of both of them. The samples were, the cells were lysed and did lipid extraction on them. So using lipid annotated, compared positive and negative modes of acquisition. In total, there was 430 lipids annotated in the positive mode and 653 lipids annotated in the negative mode. So taking these information from the cells that were harvested, etc., and doing statistical analysis, we were able to show a clear and nice separation between the different treatment in the samples. It looks like the MPA is somehow close to a vehicle treatment in the positive mode, whereas there is a clear-cut separation in the negative mode. Looking at the data now a little bit more in depth. So compared to the published published results, what you see in green are in agreement with the with whatever it was published between the diglyceride levels that the increased decreased in diglyceride levels and increased triglyceride levels. Between the LPC and LPE, we have no significant results. In this case, no significant decrease. What was interesting in this study, actually, what came up in our results compared to the ceramides, actually. In the paper, so we have increased, or my colleagues have found increased levels of ceramides, whereas decreased levels of phosphatidylcholanes. And that wasn't reported in the publication. So the idea was to go dig deeper and why do we see different results? So I'll pick the ceramide example in this case. And with the annotation, complete annotation and different classes of ceramides, we were able to find actually and distinguish that why we have differences. First of all, in the publication, they've used shotgun lipidomics, whereas we've done a little bit more in depth. And what my colleagues have found out, these particular isomers of ceramides, they are nicely separated and identified by chromatography and with MSMS with a lipid annotator. And there are slight differences between them and with the samples that were, or the cells that were treated with a combination of drug. So this is the kind of information we could get different while using shotgun lipidomics. It was nice they were able to highlight classes of lipids, but digging deeper into more profiling lipidomics, you could go a little bit more in depth and tell a particular lipid if it's up or down. Of course, in this case, you needed both good sample preparation and good chromatographic method. By looking at the same data, actually, they wanted to dig a little bit more deeper and see with the untargeted approach. So working with that, they found one particular mass that was the 339 that was significantly changed between the controls and the treated with the two compounds, the combination drugs. So they've used the MPP and lipid annotator tools. Between the Kendrick's plots and the lipid annotator tool, we were able to spot on, identify or say that this peak or this mass corresponds to a ceramide, a particular ceramide. With generation of the formula, we could tell that it could be potentially this one, the 18220. And as a matter of fact, this particular ceramide is not present in the database used. So this is why it wasn't annotated initially. So the bottom line is using all these tools can help you to get more in depth into the results, into the annotation of these lipids, regardless, actually, even if they are not in a database, particular database, it's not impossible. One of the last finishing steps for me was, I mentioned within the separation using the ion mobility as well approach. As I said, it sits well in terms of separation in terms of milliseconds compared to the chromatographic separation in the order of seconds and the QTOF acquisition speed in the microseconds. The ion mobility gives another dimension or another information about each of the analytes, which is the collisional cross-section of each of the molecules of analyzing it. And this correlates with the structure of it somehow. So working with a collaboration with the University of Graz, these are the results of Dr. Thomas Eichmann. We have created a CCS library of more than 800 lipids, and these lipids are biological lipids, so they're not in silico generated. These come from different mouse strains and samples that were used in this case. They were manually annotated using MSMS and with the all ions ion mobility for the CCS values. They were, of course, generated using 3 biological samples and validated with different laboratories with an accuracy less than 0.5% in RSD. So using this library, what we have done, or what Dr. Eichmann has done, is used a mouse model and then did extract the lipids from different tissues and try to to use and put this library and CCS library into place. So from these tissues, when analyzed with a quicker lipidomic method, about 60 minutes, more than 49,000 features were found. So if we're trying to identify them with accurate mass or even MSMS, we weren't able to do a quick identification, whereas using the CCS value, we had a very nice identification with low false positives, rate of false positives. So this gives another confidence in terms of identification for the lipids. It's I would say once you have your library or this work done, then it becomes easier to do. The PCA analysis, this is just to show you the different tissues that were used. Some of them were as well brain tissues, epidermis, lung, liver, etc. So I hope by the end of this talk, I was able to show you even a quick glimpse of the different solutions that we offer, from the strategy of the acquisition that our software allows you, the tools for the annotation and the statistical analysis, and the addition of the ion mobility as well for the analysis, and in order to get more the important part, which is confidence in the annotation of lipids. With this, I finish my slides. I hope I'm still within the time. I'm looking forward for any questions, and thank you again for your attention.
Thank you very much, Hania. Very interesting. I think we have time for one question from the audience. No, I have one. Okay, then I ask a question.
Yeah.
Since you mentioned, now I'm curious to learn more about the coupling of SFC and mass spec.
Yes.
How does it work, basically? How is the ion source? Is it any different from conventional electrospray source?
Not at all. Not at all. Actually, at least for the Agilent SFC coupling with the QTOF or with any Agilent instrument, source conditions are practically the same as the LC, and it's like mix and match practically. You just put it in front of the instrument and that's it. Actually, in my hands, it worked nicely for lipids. I've ran standards of gangliosides. I've seen that you were interested and you've done that before. So I've ran standards and I've run brain samples with SFC, with gangliosides. It worked nicely. The work was done a few years ago. I would say now with my experience, I could still do better.
Okay, very interesting. Thanks.
You're welcome.